Okay. Good morning, everybody, or afternoon, or wherever you are. It's my pleasure to lead or moderate this next field day, which will be on the FANG project, Functional Annotation of Animal Genomes. So my name is Jack Dekkers. I'm a professor at animal science, animal breeding genetics at Iowa State University, and I'm one of the co-PIs on the agricultural genome to phenome initiatives, and I'm leading the field day portion of that project. And today we're going to talk about functional annotation of animal genomes, and so we have three excellent speakers who can talk about what this project is. It's really a good example of how, in this case on the livestock side, how communities have come together across disciplines and across countries, across continents, to build a consortium to make progress in this whole area of annotation of genomes, which is of course needed when we want to go to genome from genome to phenome. So I'll introduce our speakers. We'll have breaks between the speakers. If you have questions, please put them in the chat. But also, feel free to shout them out, and if you want to. And I'll, if there are questions that are relevant to the topic at hand, that is, what the speaker is speaking about at that moment, I'll interrupt the speaker. But otherwise, we'll leave it to the end of the speaker in between the breaks, and then we'll have a panel discussion at the end also. So. First speaker is Elisabetta Giuffra. Elisabetta is originally from Italy. She's currently in Italy, but she works at INRAE in France, which is sort of the equivalent of USDA in the US. And she's a senior scientist there and has been a co-PI or a co-chair of the FANG project. And she's going to tell us about how this started and what it does, and then turn it over to, so I'll turn it over to Elisabetta.
Thank you very much. This talk, I will talk about FANG and how this initiative is boosting or empowering genotype to phenotype research in farmed animals. Okay. Doesn't go? Okay. G2P, genotype to phenotype, or G2P. Well, deciphering the G2P link is a major goal of modern biology, and predictive biology implies using genome information to predict complex outcomes, such as disease, a complex phenotype for production or whatever. And this simplified illustration is a way to represent this very well known equation where phenotypes equal genotype plus environment, plus, of course, interaction of genotype per environment. That could be complicated by other less simple, well, not simple really, but by other parameters, like, for example, how to address the microbiota for complex intestinal traits. Anyway, this concept of G2P is considered as a key for addressing challenges to achieve sustainable and responsible global food production in the next decade. In this talk, I will go through the following points. I will talk about FANG, why, the community of the core activities since start, the research phases, the next challenges we identified and baptized from FANG to fork. I will then go through a list of projects that are ongoing, first with the US projects, then the three European H2020 projects federated under the Eurofang label. I will also take the occasion of talk on behalf of coordinators of 2 new EU projects that are starting in June this year. And then I tried, well, in my view, I made my view about that, to draft the challenges, needs, and measures opportunities that I can imagine for the future of the FANG. Well, why FANG? The aim is to discover basic knowledge of genome function, to decipher the G2P link in both terrestrial and aquatic animals. And the main goal is to achieve comprehensive functional maps of genomes, useful to unravel which bits of each genome are functional, that could be repressed, activated, poised. And these, of course, should be made at different times, like during development, different cells, different tissues, different conditions, and so on. And this is key for providing information for fundamental sciences to link cell, tissue, and whole animal scale knowledge. Important, for example, for dissecting the components of emerging and complex phenotypes and genotype per environment interaction. And providing information to accelerate the genomic improvement, like, of course, genomic selection, the most important for us, which is about predicting organism level phenotype through linking to variation present in large numbers of different genomes. For example, to reduce the search space for causative variation and improve cross-population accuracy. And these concepts were outlined in the white paper published by FANG in early 2015. The building of functional maps of genomes, here it's illustrated by a quite famous ENCODE illustration, which has evolved in time. And I underline that it is from 2019, because now the website of ENCODE has changed. And I think one reason is that the number of assays is so long. I mean, the list of assays available now is so long that it's essentially impossible to list all of them in a figure. These assays by sequencing can, for example, if you look from the right to the left, from the ultimate level of transcription, so how RNA transcription is regulated and so on, up to the different levels, so to modification like post-translational modifications of instance, accessibility or closure of the chromatin, up to the 3D conformational chromatin ending chromosomes. And these assays can be performed on cell lines, as it was done also in the beginning by ENCODE, but then this evolved to tissues and primary cells in tissues. And of course, it's evolving fast also to characterize really each primary cells and understand better the complexity of tissues. And the early lessons from ENCODE were where I remind is the Encyclopedia of the DNA Elements projects, which was developed for human and model species, but followed then by several other consortia, because we talk of the first decades of 2000s. But this was followed by several other consortia that were either focused on different organisms and or focused on specific biological aspects. And ENCODE has shown many, many things, but has surely put in light the importance of promoter functionality to explain the variation in the expression of RNAs. It annotated a lot of non-coding variants, and that, of course, lie in ENCODE annotated functional regions. And it found, most of all, that SNPs associated with disease complex traits are really enriched within non-coding functional elements that, of course, we probably knew before. We knew that we look for traits that are complex, regulatory, not easy to shape and to understand. And in addition to the scientific catalogs, I mean, to this catalog of functions, ENCODE has given a great contribute in terms of understanding how a community should develop and work together. They have really driven technology for the development, for example, of several assays by sequencing and by standardization. And by doing this in OpenShare, they have developed freely accessible catalogs of regulatory genomic elements of a human genome sequence. And this has shown to have a really big impact on the human biology research. As you see, if you see in the graph, you have that in light blue using ENCODE data and violet from the ENCODE consortia. So this is an old graph. It arrives to April, June 2014. But you have, of course, a steady increase of ENCODE consortia publication, but a very sharp increase of publications of groups that use ENCODE data. And that is something that we expect to see also in other organisms, in fungi as well. In the bottom right, I put just some examples of how studies have, what studies have allowed to develop in humans and medicine. What about community and core activities in START? There has been a preparative phase led by a small group of scientists from 12 countries, 2011. We are today 535 members. We have been working to standardization, I would say quite a lot, of core assays, of experimental protocols and bioinformatic pipelines. And this has been done through discussions and fund committees. We have worked to coordinate and facilitate data sharing through the Fung data portal developed at MBLEBI. And we had also exchanges with the community and main agencies representatives. And I'm going to give the example of the GoFung workshop that was organized by Christagolin back in October 2015, where we had about 100 attendees and 6 funding agencies presented talks. In this case, there was also a report that we published in Animal Genetics. And this was the first event that really set in motion organizing call for funding for both the US project and Europe. And in fact, there were topic calls in 2017 at USDA, in 2018 EU. In EU, it was very important, but I will not be showing a slide. The meeting organized directly by the European Commission, the office of Jean-Charles Cavit in October 2016 in Brussels. That was attended as GoFung by different scientists and industry representatives from EU and US. Fung research is going through phases that are much overlapping but distinct, I would say, because of course you have first the annotation of maps, the functional annotation of genomes that are the main biological reference. And to tackle these, scientists have addressed, first of all, the main farm species that at the time initially had the best genomes assembly, with few adult animals represented. Phase 2 implies to more complexity. It means that these annotations are performed considering different developmental stages, physiological conditions, environmental perturbations, and of course, additional species. And it really enters more in the genotype to phenotype for the implementation of predictive biology, because it tries to tackle larger populations, so more animals and new breeds, phenotype populations, and of course to involve industry. The last phase, it's the from Fung to 4, and this is to match the challenges of sustainable production for next decade. I come back to this. On the first phase, I want just to cite our involvement with pilot projects, and there were others also on different species, but I think we were the first in France and the US. So at IRAE and at UC Davis, 2 projects coordinated by these 2 institutions. In the case of FrageENCODE, that was the French project, it is still a federation of 14 research labs and animal facilities, because ERA has quite a number of experimental facilities to raise specific breeds and populations. And it was focused on the annotation of 4 target species at 3 target tissues by a number of core assays. And we produced some papers, including long non-coding RNA papers, like especially produced by the unit of PEGAS in Rennes, which is in Brittany. And here I paste the main publication by the UC Davis Consortium, which appeared earlier this year. These first 2 phases have really fed the US large-scale projects that started in 2018. I underline that there are several fund-related projects around the world, and they are increasing, but for sure in Canada, Australia, China, and others, that I cannot really tackle in this call because there is limited time and it's also difficult to know exactly everything. And as I said, 2019, we have the large-scale project start, and there is the US, which is wrong there. Fund to fork, the start is 2020. Fund to fork, or next challenges, this is the figure that is also in the paper in Genome Biology by Clark et al. The challenge is to link G2P in diverse populations of animals. For example, you may have large phenotype collections and the new populations of animals raised in well-defined environment, like indicated at the bottom right. It's to apply innovative new technologies to model G2P at the cell, tissue, and whole animal scale, and of course, to connect this information. I'm particularly interested in organoids and induced pluripotent stem cells that are being developed in some labs worldwide, that represent the species, breed, and population diversity, and really making repositories of these animal resources, and can be phenotype in vitro or used for validations of specific causative candidate variants. And they go together with high throughput in vitro system, always for G2P, where you can combine in vitro, these in vitro models and genome editing, as in organoids. And I will mention also the single cell atlases that are really a very, very important addition to understand the genotype to phenotype relationship, to face the complexity, the heterogeneity that is in the rough tissues. And that, of course, is important also to characterize these models and to and impact on all the other methodological priorities that you see here. Another aim of Fund to Fork is for sure to reinforce the interaction with industry. I would say that reinforcing the interaction with industry and stakeholder is really a main goal also for the novel program of the European Commission, which is Horizon Europe, and that is starting this year in a matter of months, I think. And the idea in all this is to devise wide, large-scale infrastructures to build governance for FUNG. And I will come back to this, but the aim is to exploit, harmonize, and reinforce the efforts of the different communities. I start now to give an outline of the 3 main US projects that with whom I've been interacting the most, especially for the first 2. The first is about the functional annotation of the porcine genome. It is coordinated by Chris Taggle, Iowa State University, with colleagues listed there in the slide from UC Davis, Michigan State, USDA, and ADC, and Mark. And the goal of this project is to catalog functional elements in the porcine genome for many biological states and to create chromatin state maps. It's focused on the epigenetics of 25 tissues. They have an interest in the genetic control of gene expression, using also fetal tissues from genetically divergent crosses and correlated ASE and chromatin modification effects. They have a focus on the immune system in pigs, the circulating blood cells, and more recently, working quite a lot with single cell analysis of PBMCs and immune tissues. And of course, they develop data integration of data produced by molecular assay as core assays of FANG used by many projects now. For the bovine, the coordinator is Pablo Ross from UC Davis, with several colleagues and several institutions, but I'm not going to list completely, with 4 main objectives, which are here in this figure, on the transcriptome annotation and chromatin state annotation, data integration, and also the integration of this information of functional maps with genome-wide association studies for typical production traits. And as biological sample, we use adult tissues, mammary tissues, and also fetal tissues, plus culture cells and embryos. And finally, the genome-wide annotation of cis regulatory elements in the chicken genome is the Chicken Annotation Project of the US, and it is coordinated by Yvonne Dreschler in Western University of Health Sciences with David Hawkins at the University of Washington. And they have a very focused aim on immunity traits in the chicken. So the cells and target tissues they target are really relevant to this biological question, together with questions about reproduction. So the female reproductive tract, ovaries, oviduct split into isthmus, magnum, and shell gland. And the assays used are, again, let's say the typical core assays. Eurofang, EU. So Eurofang is essentially a federation of 3 projects that you see in the middle. And the idea of Eurofang is to leverage functional genomic information to make animal production more robust and sustainable in Europe and beyond, because of course we all have the same big aims at the end. The specific research aims are quite well-known terms: increase efficiency through precision breeding, increase disease resistance, minimize environmental impact, and using joint strategies on communication and dissemination, training programs, and of course, sharing research methodologies. These are the key European projects ongoing since 2018, where we spent 4 years for 6 million budget each. And they have common point between them. They are focused, of course, on functional annotation, but also they dedicate considerable resources to how using these improved annotations. So they have different tasks of scope-oriented research where typically larger cohorts of animals are used. And the other commonality is that all include a consistent effort in dissemination, training, and networking activities. The projects have you will see they have their website, of course, and so on, but they are also listed on the NBLEBI FANG data portal. And about Eurofang, I will mention this workshop that was held in Inkster, so at NBLEBI in February 2020, 1 minute before the lockdowns everywhere, basically. It was organized by Peter Harrison, who is actually also the coordinator of Eurofang. And it is this workshop that really launched the Eurofang concept, because it combined 20 key scientists from each of the 3 projects to develop shared coding standards, foster collaborative work on pipeline development. And in fact, following this in November, Bob Reg workshop, the project Bob Reg organized a workshop on NextFlow and FCore that was attended by participants of all these projects, to develop comparative analysis across the different resources and methodological possibilities offered by the 3 projects. And I mentioned that the working group is now meeting regularly, organized by Dan McQueen at Roslin Institute, where we discuss, look at our data and the opportunities and discuss on how we could pursue some common study. And then we develop a shared training and shared dissemination strategy. And this is going on and in progress with Chagla Kaya from EFAB and EAAP collaborators. Very briefly, I will go through the 3 projects because I wanted to save some time also for showing you the new projects. But I will go briefly also because you see you have for each project the website, the Twitter account, and most of all, we communicate quite a lot. So this information can be found. So Aquafang is about European aquaculture, advancing European aquaculture by genome functional annotation. It's coordinated by Sigbjørn Lien. Bovreg is on the bovine to regions that are relevant to phenotype diversity and plasticity in cattle. The coordinator is Christa Kuhn in Germany. GeneSwitch is on the regulatory genome of swine and chicken. Functional annotation, especially focused during development, plus also in this case, a pillar for implementing the new annotation for basic and applied science. It's coordinated by me with a co-coordination of my colleague Hervé Aklock. The new projects that start are really starting now in June. And these 2 persons who are Frédéric Pitel and Eric Payou kindly said, okay, let's present our projects. And I think it's very nice because I hope we will join Eurofine pretty soon. One is about chicken and pigs, again, Geronimo. It's about the genome and epigenome in hybrid breeding in monogastrics. The other one is Rumigen in ruminants, especially in bovine. It's towards improvement of ruminant breeding through genomic and epigenomic approaches. These are really follow-up of the first calls to which we replied, which in my opinion arrived a bit too early, as also the officer recognized, but here was the occasion and we are happy these projects, big projects are starting now. Geronimo, the aim is to use knowledge on genome sequence and its regulation and expression to analyze the epigenome in relation to efficient livestock production, develop tools to improve breeding schemes and ensure optimal genetic and epigenetic diversity, with development of methods, tools, and models to assist industry and policymakers. I put here the overall concept, and I will otherwise skip a lot of details because I wanted to finish the call in not so long time now. But it's clear from these approaches, and you will see this here, but also in Rumigen, that there will be really, by definition, important inputs from GeneSwitch for pig and chicken, and from BobGen for bovine, as a continuum of the research in Europe, but well connected to the international one. And that somehow these projects, for the keywords they have, like environment, sustainability, quite at the center of them, and resilience and so on. I mean, in the epigenomic traits, really epigenetic traits induced by environment, they are really going to enter in the next Fang2/4 phase. Is the consortium. This is about Rumigen. I guess you will find the back of the slides, so I will go quite fast on it. And here I wanted just to end with what I see as challenges, needs, and measures, opportunities for the futures. Well, I tried to organize my thoughts. This could be surely improved. I thought that challenges in Fang, since always, is that there are several species, so several genomes with diversity of research aims and approach, and that the key, the measure on the right, is to keep reinforcing the central support infrastructure and spread really the adoption of experimental standards and open sharing. but also take the occasion to not see this like a weakness in the fragmentation, but also think that there are new scientific opportunities because there may be new ways to approach biodiversity studies, the study of agricultural ecosystems, new comparative genomic studies, and so on. There is a need or an aim, continuous aim, to enlarge the scientific community and diversify actors and stakeholders. And here, I think the new paradigm for research is that now research should be research plus communication plus training of a novel generation of scientists, never miss one of these components. And as well, an opportunity is to strengthen the connections, some already exist, to parallel consortia that use emerging genomic technologies and can avoid a lot of time in optimization and so on for us. Then a problem that we have, should have, or exist in my opinion, is that several groups, network, consortia aim to the same goals, although from different angles. And I think we all know that competition is strong for limited resources. I think the opportunity is to harmonize efforts as much as possible on the basis of cooperation and fair competition or something like that. I'm citing Mario Draghi here from G7, but anyway. And the measures is that we should outreach and work effectively with new consortia. And right now, FANG is paving a collaborative scheme with the FarmGTEx consortium, for example. Finally, I think the most important one is that it is difficult, let's say impossible, I say, to fund unified and multi-country projects. And thus governance, that is this essential need for FANG, is not yet adequately supported. And here the opportunity, I think, is to really design country-based but well interconnected infrastructure. And this is what the US are doing, I know, and what we are trying to do in the EU as well, and possibly other countries will be aiming at these goals. And in the EU, Emily Clark, with me and members of the FANG steering committee are preparing EU-based infrastructures. I will not go into detail, but you see here that in this ambitious large-scale infrastructure, there are many of the elements that I presented today. And I thank you a lot for your attention, and I end here.
Okay, thank you very much, Elisabetta. Very, very impressive presentation, and what FANG has been able to accomplish and has continued to accomplish. And it's interesting to see how different projects have grown out of FANG, and it's really been instrumental, I think, to leverage additional funding, both in Europe as well as in the US. And I think it's a good example of what we also want to accomplish with AG2PI, but now going across livestock and crops. Anybody have a question? I don't see any questions in the chat. Anybody have a question at this point? Otherwise, we'll go to the next speaker. There will be questions at the end also. Okay, well then we'll go on to Peter, and but yeah, Elisabetta will stay on, and we'll have a panel discussion at the end. So Peter Harrison is our next speaker. He's a genome analysis team leader at EMBL, the European Bioinformatics Institute, and he's been very involved in developing that data infrastructure for FANG and the heterogeneous data that it generates, and making that available to the community. And so, Peter, turn it over to you.
Thank you very much, Jack. So today I'm going to talk, to follow on from what Elisabetta was introducing around FANG, about the importance of rich, fair, validated metadata, the standardized protocols and analyses, and open access data sets for GTP. So I'm going to focus on examples from FANG, but really this is applicable to any large-scale genomics consortium where they're doing genome to phenotype research. So as Elisabetta highlighted, the Global FANG Initiative really consists of hundreds of researchers across multiple projects spread all over the world. And this creates a real challenge from a data coordination perspective, because it's still key to ensure that all of the research generated by these individuals is comparable, both within species and also across species for larger comparative studies. And really, when we're talking about genotype to phenotype research, we're really requiring quite rich, validated and standardized metadata in order to be able to interpret our results. But also the data sets themselves really need to be high quality, and this requires standardization on the data generated, but also on the analyses being performed and tracking what analyses are being performed to ensure that this is reproducible. So this is where the Data Coordination Center for FANG comes in, and this is what I've been working on for the last 5 years at Embley BI. And really, this is to support FANG's aim to produce these high quality functional annotations of animal genomes and the creation of this rich genome to phenotype resources by really ensuring that the FANG community has really richly described data that's available and easily searchable, that we're really consistent in the way that we report that data and present it to the community, that we really facilitate the members of the community to try and standardize their sampling protocols, the way in which they're generating their data in the sequencing centers, and then also the analysis that they're doing on those data sets. And also really facilitating FANG's core aims from when it was began of really being open about pre-publication data under the Fort Lauderdale and Toronto sharing agreements, ensuring the data is reusable for other data sets, just as we saw with the ENCODE graph that Elisabetta showed, that there is a huge amount of research that can be done additionally after the main papers are done on a consortium such as this, but also ensure that these cross-project analyses and cross-species analyses are possible by taking these steps. So really what makes the FANG collection special, in my opinion, in comparison to other data that you may find in the archives, is really this focus on really rich, consistent, and highly validated metadata descriptions. So with a standard submission, you may have to provide the organism and maybe some information about what was collected and what you've done with it. But in FANG, we've really tried to insist and mandate that you really must provide a significant amount of information about the sampling that you're doing, the data sets you're generating, and the analysis that you're performing on those data sets, so that we're really producing a high quality product for the community when it comes with a FANG label. And this is also about, within FANG, we've standardized on a set of core assays that does evolve as technology has improved. We are incorporating more assays into the FANG umbrella, but ensuring that these are applied uniformly across projects, because unlike ENCODE with a single funding resource, FANG is made up of multiple projects with multiple funding agencies spread all over the world. So we need to try and, through the steering committee and the working groups, agree on the way in which we're going to conduct our analyses. So connected to this, we have mandatory sampling and experimental protocols, which give a really detailed level of information about the sampling that's performed and the analysis steps that have been performed on the data sets. And these are available alongside the data sets so that you can really understand what has been done to generate this data. And really in this sort of next phase of FANG, in this current phase 2, we're really focusing on moving towards standardizing the analysis that's performed on each data set, and sort of trying to encourage groups around the world to use similar analysis platforms, or at least report really clearly about the analysis that they're performing. And really just to produce a data platform that really ensures that we're providing open and fair data. So that's data that's findable, accessible, interoperable, and reusable. So for FANG, in the early stages, there was a metadata and data sharing working group that was set up to really try and think about all of the different sampling, experiment, and analysis questions that you would need to ask in order to make a study reproducible and studies comparable if they were done by different groups all around the world. What we came up with is that actually you need a huge amount of different metadata questions in order to cover the breadth of different analyses that are performed within the FANG umbrella. So this is both sampling in terms of the different organisms that you're going to be working on, and then the generation of cell lines or use of embryos. Within the experimental range, we have a huge different range of assays, from RNA-seq to CAGE-seq to ATAC-seq. And all of these require different specific metadata to be captured in order for the experiments to really be useful. And this is constantly evolving with the community. So we are constantly updating this metadata and versioning this metadata template for FANG. And recent significant changes include the incorporation of aquaculture into FANG for the first time with the Horizon 2020 project Aquafang, single cell sequencing becoming much more common amongst the different groups, and also a sort of deeper focus on really clearly defining developmental time points and making those comparable across species. And this metadata standard is really trying to aim to be really easily understandable by those that are using it, be highly repeatable and reusable, And that really focuses on the consistent use of terminology and being really structured and specific with what we require. So to really try and standardize the terminology, we use ontologies wherever possible to try and quantify and standardize the way in which we're having descriptions. And really, this is really trying to just accelerate the research that can be done by enabling others to really easily understand all of the various steps that have been taken on in a given data set. So you can see in the snapshot at the bottom here, an excerpt from our metadata standard, where you'll see we're catching a huge range of different levels of information with clear descriptions, different levels of mandatory requirements, and then also specific ontology sections that are required. So we're really quite specific about which ontologies you're allowed to use within the FANG metadata standards. And really within FANG, we recognize, as most projects do, that metadata isn't equal. But what we've done is go beyond just the standard mandatory or optional distinction and really try and include a middle ground as well, which is a recommended area. So instead of, when you look at a consortium as large as this, it would be very difficult to have everything as mandatory. We've often found that there are a few exceptions that would otherwise cause issues if you were trying to that data would either not be able to be submitted, or you would have to lower the quality of the standard for everyone else and by making not everything mandatory. So we have this middle ground, which is recommended, which is really data that we really see as highly important to the project, but we accept that not in every situation is this possible for someone to collect. But we've also tried to control the terminology used to describe why you can't provide data. So you actually also have to provide one of these 4 controlled terms to say why you weren't able to provide a particular piece of information, so that it's really clear to those coming and using your data sets why that data is missing, whether it's that it just simply wasn't possible to collect, or that you're potentially they can contact you to have it, or the fact that it was restricted access for some reason. And FANG also has a real concept of legacy standards. So there are a huge range of data sets which are really useful for doing your GTP research that have been done outside of our consortium. So we've actually tried to assess these and to bring in data sets into our portal, and which will support our researchers in being able to do their analyses, but they might not actually be able to meet the full FANG standards. So we have a separate legacy standard upon which we assess other data sets to pull into our portal that will hopefully support and provide additional data sets for people to use for their analyses. So the FANG data model really focuses on trying to link all of the data that is generated within the FANG projects together. So it starts at a really detailed metadata description of the organism, including the relationships of that organism to other organisms that you may be sampling, the sex, the location, specific coordinates. Connected to that, you then have all of the specimens that you're taking from that organism and perhaps generating cell cultures and cell lines from those specimens as well. And these are all linked together with biosample relationships. We then, from those samples, have all of the assay data that you may be generating, the raw primary data from RNA-seq and ATAC-seq, CAGE-seq on these data sets, all linked through to these samples that you can navigate and collect all of the data obtained from a single individual organism. And finally, we also have the analysis data that's generated from this primary data, all linked back to that original organism, so that you can really clearly see what data has been generated and what analyses have been generated from which raw data. And so you can go back and reanalyze or look at the phenotypic information to really clearly see what's been done. So the other thing that we have is a mandatory requirement for detailed protocols with every submission. So part of our metadata standard has protocols, PDF protocols that have to be provided along with a submission. So within the FANG portal, we have a protocol upload area where you have to prepare PDF protocols on sampling or on the primary analysis that's being performed. And also, as well as being a sort of a real aid in the reproducibility of these studies, it's also actually a really useful resource in itself for those that are preparing to do FANG-like studies in the future, because it's a useful reference for how people have done particular analyses. So how has that group in Germany done their ATAC-seq? You can actually go and read their really detailed protocol on how they did it, and then also will then encourage others that follow to follow the same protocol, and that data will be more comparable when it's generated in the future. So it's also encouraging standardization across the partners, and we actually have working groups that have been discussing and agreeing on the best way to perform particular sampling methods or the best way to perform particular analyses. So the metadata itself would be no use if people didn't follow it. So part of the Data Coordination Center is to really ensure that all of the partners are meeting these standards, and these standards are not easy to meet. It's a lot of work for a submitter in order to meet these standards. So all of the data goes through a pre-submission validation step that's within the FANG data portal, and you're not able to submit until you're fully compliant with those standards. So it actually blocks submission until you're compliant. So from a user's perspective, what they're filling in are metadata Excel templates. So this is the easiest way to gather information from a range of labs who may be working on a particular experiment. And the validation service will take this and convert it and validate it and really highlight not only the errors, so when you're not meeting the standard, but also warns on suggested improvements, such as being more specific in an ontology if it believes that you could provide a better term. But what we've also done is then brokered the submission to the underlying archives. So in the past, in the early stages of FANG, you'd validate your data and then have to go off to each individual archive, so biosamples or ENA or GEO, and submit your data there. But what we have now is we have a brokered submission service, so it handles all of that on your behalf. So you submit your data from within the FANG portal, and it will submit it to the underlying archives on your behalf. And as well, we have this requirement for mandatory protocols, as I've already mentioned, and we have a very active support desk to try and help users to submit their data to FANG.
Peter, maybe I'll interrupt here because there's a question in the chat that I think relates here. Question from Jean-Luc Janning. He asks, are funding agencies requiring researchers to document and submit all of this metadata? What help are researchers getting? And because it is a lot of work. So is it a carrot or a stick that gets researchers to submit this data and fill all this information in?
So I think the funding agencies, I mean, are in some ways requiring this because if you've applied and you've obtained funding under a FANG umbrella, so for example under the Horizon 2020 projects, these were specific FANG calls, so you had to agree and comply with the FANG standards. So you were agreeing that you were going to submit your data to FANG. So it was a fundamental requirement for those 3 Eurofang projects that they would meet the FANG standards with the submission of their data. So they were requiring this, and it was factored in as part of sort of the plan for each of these projects that they were going to have to do this submission, which is actually why we as a partner within these projects have really worked to improve that validation step. So as part of Eurofang, we were funded to improve the sort of the validation and submission experience for the Eurofang projects, which is why we now have this brokered submission service, which is much easier to use. But we will freely admit this is a lot harder to do than just submitting to NCBI or ENA just on your own bat as a researcher, where you may only be required to provide very basic level of information. But really, as a community, the value in this is that we're really generating a resource which can truly be seen as really high quality. So when people see FANG data, they will know that there's this huge range of metadata associated with it and standardization of the analysis and the protocols that go alongside that. So I think that's what as a community, as a FANG community, what we really see in the value in that.
Okay, thanks.
So in terms of ontologies, this is one way in which we're trying to standardize the metadata that we produce. So we're really trying to use these ontologies as they have a unique identifier and group all of the different ways in which you could particularly describe a particular piece of biological information. And importantly, these ontologies have hierarchy, which allows you to really, when you come to do search on data sets later, you can then group together, for example, all of the terms that were specific for different parts of the brain. You could do a search for a brain and actually obtain all of those specific parts if people had used more specific terms in their metadata submissions. But one of the issues we have with ontologies often in agriculture genome to phenome research is that many of these ontologies, and there are exceptions to this, there are some great agricultural specific ontologies being developed now, but many of them sort of have descriptions or terms that preclude their use in agriculture. They're just simply written from a medical or a model organism perspective, and just means that they're just not very useful for agriculture. So one of the things that we're working on at the moment is for FANG is an ontology improvement service that's really going to help the communities crowdsource improvements to these ontologies. to try and drive this forward. And there's been, over recent years, some great workshops where people have really focused on individual areas to improve ontologies, but this is trying to put in place a service that people can use to run focus sessions like this, but it's also more generally come along and provide, where they have specific knowledge, improvements to ontologies. So what this will do is it will gather all of the terms that are important for the different species areas within FANG. It'll allow you to flag those that you just know to be wrong or incorrect. And then also, if you have that experience and that knowledge, you'll actually be able to provide a better term or a better description. And then we will then, the community will then assist and vote on those. And if enough people think that that is an improvement, it will then get forwarded to the underlying ontology, as many of the ontologies are now in a position that you can automate some of the reporting of ontological improvements. So that's sort of the aim of this service that we're going to be releasing in the summer. But also, although this is focused on FANG, there is sort of no reason why the architecture behind this could not be applied to crops as well in the future. So really, the next sort of area of standardization comes into the analysis. So moving away from metadata when we're actually coming to process the data. And there's sort of two streams to standardizing the way we perform analysis. There's centralized standardized annotation platforms, such as Ensembl at EBI, which are producing a sort of a standardized annotation pipeline for crops and animals, pushing all of the data through a single pipeline to produce a sort of uniform and comparative resource for those species. But then probably more importantly for sort of the individual groups within FANG is a desire to have all of the individual projects and researchers using common analyses or putting together analyses that can be considered gold standard for use within FANG. So we're really trying to ensure that the pipelines being generated, and there's lots of discussion going on at the moment, are as reproducible and comparative as possible. And there's particular focused efforts upon this that I'm going to describe in a moment. But we also have real opportunities with the advent and the rise in popularity of cloud analysis platforms to have some of these pipelines, once they're agreed upon, pre-installed alongside the data sets. And we have perhaps opportunities at Embassy, which is in EBI's cloud, but also a lot of the biological effort that's going into Google's cloud, but also in the US, Cyverse and KBase that may be able to have many of these analyses available to their user base ready to go. So sort of the vision really for the standardized development of analyses in FANG is to have all of the projects contributing to a single set of analyses. This isn't going to happen. There will always be individual branch points and individual situations where people have requirements that go away from this. But at least what we're trying to do is to have development based on our same principles that we have for our data, to really have principles of open science in terms of open source code development, and really ensuring that everyone makes their workflows available and as reproducible as possible if they can, so that people will then contribute to the same pipelines rather than just reinventing the wheel every time that they come to do an analysis. So really, we're trying to develop an active and collaborative bioinformatics community within FANG. And we have a particular focus at the moment within the 3 Eurofang projects, so really exemplifying this approach as they're within these working groups that we established at the meetings that Elisabetta described. We're really trying to coordinate on our approach to bioinformatics analysis within these 3 projects, but also with input from US and Australian projects as well. And really, this is the technology that we have to be able to do this is widely available, and many people now are implementing many of these in the development of their pipelines. So we have workflow managers that can really easily make these constructing pipelines and repeating pipelines easily manageable and also scalable. We have containerization technologies, which allow pipelines to be run in identical environments wherever they're run on different cloud or local computes. And then we have the cloud technologies to ensure reproducibility by sending the analysis to the cloud rather than moving the data around. And this is really to try and sort of a future model for how we can really accelerate and lower the cost and how these analyses are going to be performed. So really a future vision, which we may get to one day, is that the FANG bioinformatics working groups and projects and community contributes to a single set or a range of different pipelines which are agreed and evolve over time. And these are sort of a master record of approved pipelines that the community regularly uses. And that these are then passed out to the different cloud or local, and you can utilize them on your local university compute as well, to run your analyses in a common and standardized way. And two of the efforts that are working towards this, we have our own FANG GitHub page where people are contributing their pipelines they're working on within different projects. And there's quite an active discussion on some of those pipelines. But we also have the NF Core, which is a community effort, really being used very extensively within the Eurofang projects to collect and create pipelines within an Nextflow management environment. And these are Eurofang is agreed to use and contribute to NF Core wherever possible when they're doing their analyses, so that we have a range of different pipelines that are being developed collectively by these three consortia, FANG consortia, and working on them to produce a reproducible and common set of pipelines. And this also then contributes to the aim of these Eurofang projects to be able to compare and produce comparable data sets across their three projects as well as within them. And one of the really key parts of this, even if you can't use the same pipeline as someone else, is to actually ensure that we link which pipeline you've used to the data set that's generated. So this is to really try and make sure that with your data set, you're also providing which pipeline you used to generate it, which configurations you used when you ran that pipeline. So GeneSwitch is a sort of a model for how to do this in an open way with modern cloud analysis. So as soon as the samples are taken in the field, the metadata is submitted straight to biosamples. As soon as the raw data is generated, they were submitted straight from the sequencing centers to the European Nucleotide Archive at EBI. So the data, and then that flowed through into FANG. So the sample and raw information was immediately available under Fort Lauderdale and Toronto agreements in the FANG data portal. This was then synced to the Embassy Cloud environments, and then the Nextflow pipelines that GeneSwitch were using to analyze their data were pulled into this same environment so that they could then run their analyses. And then once these analyses are complete, the data sets then also pushed into the European Nucleotide Archive and immediately available through FANG and GeneSwitch. So this is really an open way of running their FANG pipelines and pushing information directly from the cloud into the FANG analysis platform. And it's really a way of unifying the way in which we run these analyses. So on another track, another tack of important area for FANG is to support community annotation. And one of these is a way in which we need to advertise and make it easier for individuals to provide tracks that can be mounted onto the Genome browsers at Ensembl and UCSC to be able to show individual data sets that you've generated and your annotation views upon those. So the TrackHub registry is a service at EBI, which is aiming to sort of provide a way to really provide accessible and centralized way to provide these TrackHubs to the community. And FANG is going to heavily use this registry in order to advertise its TrackHubs. So a new version of the TrackHub registry is coming out in the summer, which is going to have improvements that are going to help FANG to submit and to make available these tracks. But there's also a need for greater support for community annotation tools. So we're also supporting Web Apollo, which many communities, and particularly I know in the crop world as well, are using to feed community annotation into our resources at EMBL EBI. And this is another important area for us. So all of this data flows through to the FANG data portal, which really aims to be a single access point for all of the FANG metadata and data that's generated within the project. And it has automated identification of data sets and the publications that are then used upon them. It provides all of the access to all of these detailed protocols, and then provides direct download access to the data sets. And really what we're trying to do is have completely fair data sets where every data set has all of this rich metadata, all of the protocols associated with it, links to the primary analysis metadata and data sets, then also links to the pipelines and the configuration files that we use to generate those data sets, all the publications gathered into a single location that were generated from those data sets, then also these optional TrackHub links so that people can then provide additional presentation layers from the data that they've generated within FANG. And also, as well as sort of from following Ag2PI, I've seen there's a huge amount that we can learn from each other in the animal and crop world. We also have a lot that we can continue to learn from human and model organism research. And so many of the pipelines that we're developing at EBI have been honed from our human and model organism knowledge. And we have a lot to learn from ENCODE within FANG, and we have a lot to learn from the Human Cell Atlas project for the agricultural single cell atlases that we're hoping to develop. But we also have a lot of common statistical and infrastructure advantages, such as the current moves into graph genomics, which we need to collaborate and benefit from within these agricultural research. We also have a real importance to train the next generation of scientists. This is really at the heart of MBLEBI's mission, and also is really key for FANG. So we're generating at the moment a new working group to try and coordinate and promote agricultural training, genomic training within FANG. And the Eurofang projects have committed to open all of their planned training to FANG partners. But also MBLEBI is offering a range of free in-person virtual on-demand training courses. So we had one recently on ChIP-seq and ATAC-seq that was attended by a number of people from the FANG community. We also have a range of crop resources as well. And I just wanted to briefly at the end introduce the fact that MBLEBI is really taking its commitment to agricultural research forward. So we've appointed recently 2 new groups to focus on the research support for genomics to improve crop and animal agriculture. So this is my own team, which has just recently started. It's focusing on animal agriculture, aquaculture, and biodiversity in the Tree of Life projects. But also Sarah Dyer, who's our crop genomics specialist, who's just started a new team with a particular focus on crop and metazoa and pests for genomic research. And really collectively, we're trying to produce and enhance resources centered around the work, sort of our Ensembl genome browser. And together we're going to be jointly addressing sort of some of the major challenges facing agricultural research in supporting multiple breeds and strains, graph genomics, single cell sequencing atlases, and also dealing with the scale of genomes that we're going to be coming into these browsers in the near future. So Ensembl is a genomics platform for enabling and accelerating downstream science. We have around 300 vertebrate model organism genomes. We have separate portals for plants, metazoa and fungi. But we're currently redeveloping our browser, and this will combine all of these species into one single resource and one single browser. And it collects all sorts of information on annotation and comparative genomics for regulatory data. We're supporting multiple breeds and cultivars, and this is a particular focus for us at the moment. So we have, for example, multiple pig breeds and multiple wheat cultivars available within the browser. And we're also looking at how we can use graph representations and the scalability to improve our genome browser for the future. So within Ensembl Plants, just to highlight some of our resources for crop communities, we have 94 plant species that have this genome assemblies, a range of community annotations and variation data and comparative genomics. And particular focus in these is on rice and wheat with our research projects at the moment. But we also have a lot of information on metazoa, on insect pests and disease vectors and model organisms, and probably some of the key crop pests that many of you are working on. So really, just to summarize, in terms of data coordination for genome to phenotype research, we really require this rich, validated, standardized metadata, really detailed protocols and analysis pipelines links to every data set, so you get this full reproducible picture for any given data set. Standardized and containerized analysis platforms and pipelines so that people can then benefit from the analyses that you've been performing, and really focusing on this FANG model for an open collaborative model for data reuse, focusing on FAIR principles. And then in terms of my own company's commitment to try and develop coordinated solutions for these future challenges for agricultural research, both in animals, crops and aquaculture. And I'm continuing to look forward to the collaborations that I have with European and US researchers, both within FANG and hopefully developing with AgTPI as well. So I'd just like to thank, in particular, Alexei and Akshata, who are the main FANG developers at MBLEBI, and Paul Fleechuk, who's our Associate Director and Head of Ensembl, the Ensembl team, but also many of the Eurofang collaborators from Aquafang, Bovreg and GeneSwitch that fund our research. And as Elisabetta highlighted, this was our photograph from the last time we were able to all meet in person just before the lockdown in February of 2020. And all of our research is funded by the Horizon 2020 funding for Eurofang.
Okay, thank you, Peter. Very impressive again, what has been accomplished and what's been developed. There's a question in the chat which Bo I'll ask here, and then we'll go on to the next speaker. Question from Chris Tuggle is question is whether the FANG community data resources that are being developed or have been developed, how pioneering are they, or has something similar already been developed on the human genomic side?
I mean, I think it is pioneering in the quality of the metadata that we're producing. I think actually the FANG metadata set is one of the richest metadata description sets that I've seen. There are phenotypic, huge phenotypic studies going on in human as well, which have immense power. But I think in terms of just the average scientist submitting their data, the metadata standard we put together is particularly detailed. But I think really with all these things, the sort of the funding scale that we have on the human is sort of massive in comparison to agricultural research. So in many ways, it's what we can learn from their methodological developments. And also, but we have the complexity to deal with in terms of we can't just simply pick up many of these techniques and methodologies and apply them in agriculture. We've got all sorts of complexities around applying many of these single cell atlas techniques, for example, within agriculture that are going to take a lot of methodological development by different groups to perfect. So I think although we can learn a lot, I think we're going to have huge challenges to try and face some of these.
Yeah, and of course the challenge of having to deal with many different species and breeds within species. So. Okay, thanks, Peter. Yeah, if there are additional questions, we can leave them to the end. Last speaker is Christine Elsick. She's a professor in both animal sciences and the plant sciences at the University of Missouri. And she's been very involved in the FANG project, building infrastructure and for both animals and plants. And she's going to talk about that as well as some new ideas and some new proposals that they have developed, that she has developed with others. So Chris?
Great, thank you. So I'm going to talk about two topics today. An ongoing project, which is called FANGmine. And I'm going to just give a little bit of background on the project and then talk about how it's related to some other projects in other communities. And then I'll also talk about a community effort to seek additional support for cyber infrastructure and animal genomics. So FANGmine is a project that's funded by the National Science Foundation, and it's the objective of it is to integrate data, genome annotation data from various sources and FANG functional annotation data to allow researchers to search through it and to be able to create customized annotation data sets, merge with their own data sets. And so we have several different data sets from external sources, and in the current FANGmine release, we now have some actual FANG data. So the organisms in FANGmine they're not all FANG species right now. So they're domesticated animal species, and they were all species where there were whole genomes available back when the proposal was written. And these species are of interest to individuals who are listed as members of the FANG consortium. And since this is a National Science Foundation project, we really had to kind of branch out to a wider range of organisms because NSF doesn't specifically support USDA animal kinds of projects. So we branched out, and we have other species such as cat and dog, in addition to the farmed animal species. So this is the FANGmine homepage, and currently we have two versions of FANGmine that are publicly available. We just released the second one, version 1.2, which now includes some of the FANG data. And so I'll just talk a little bit about that. This is the homepage once you enter into the FANG application. At the top of the page, there's a menu that allows you to get to different search tools and then how to look at all the data sources. And then here's a few tools in the front of the page that you can just get some quick searches, keyword searches. In the middle of the page, there's this bar that goes across that actually has categories of queries. So each one of these is a query template where if you click on it, you can get to a little menu that allows you to do a query. So here on this busy slide to the left, I show just the sort of the data model for FangMine, and just to show that there's a lot of data and it's very complex. And we do a lot of work to integrate all these various data sets. So a lot of the work that we do is really just making the data sets compatible with each other so that we can integrate them in this database. And so for that reason, we create these query templates so that people don't have to actually know about the data model. And so here's an example of one, a simple one, where if you wanted to look for all the FANG-ChIP-seq experiments for an organism, there's a menu, pull down menu, lets you select an organism. And then you can just click show result and you get an output. Over on the right shows the query builder that allows you to actually change the query if you wanted to. So if people had, once people know a little bit more about it, it's actually possible to change these queries or develop your own queries using the query builder. The output is actually very flexible. It's all tabular, and it can be modified in many ways. So you can sort by different columns, and you can filter, you can add columns, and you can also export your data as tab delimited files. So another way that you can search FangMine is by using this thing called a genomic region search. And so this allows you to select an organism and select different features that you want to search. And so if anybody's has used FangMine before, you'll notice now that the region search is a little bit different than it was before, because when we started incorporating FANG data, we wanted to allow people to be able to search through different specific FANG experiments. And so currently our only FANG data is actually from a horse ChIP-seq experiment, histone modification mark. So if you select horse as the organism and the assembly gets automatically selected, you can open up this tree to show genome features and histone modifications. And then the histone modification shows all the experiments. These are analyses that are available. And so you can select which ones you want to search. So there's 8 tissues and 4 different marks. You can select exactly what features you want, upload some positions in the genome, and then conduct and search. And so in this example, just some SNPs from a previous publication were uploaded. So these are SNPs related to morphometric traits. And in the results, there's a summary of results where it shows you exactly which histone marks these SNPs are found in. And so we actually have assigned identifiers to each individual histone mark that we downloaded from the FANG data portal. So we downloaded the bed file of the peaks, and so we assigned the marks identifiers so that we can have those in the database, and so people can search for them individually, and you can identify different features related to these marks. So for example, some of these SNPs were found in a few marks, and then we also show some genes that are nearby. And so this can be downloaded as a tab delimited file, or you can actually save the results as a list and then use that list in further template queries in FangMine. So another way to search it is just to upload a list of things and use that list of identifiers in different queries. And so this just shows uploading a list of chicken gene IDs. You get to give your list a name, and you can save your list. And then once you have a list, you can do different things. You can use it in different queries, and then you can also, for genes, you can do different kinds of enrichment. So that was a very kind of quick overview of things you can do with FangMine. We're actually planning on doing an online tutorial next month, and we'll announce that on AngioDynMap. And that's really all I wanted to say about FangMine, but I wanted to point out that our work in FangMine is related. Actually, it kind of branched out of some work on some previous projects that we worked on. So my lab has pretty much always been involved in two different communities. So we've always been active in the livestock research community and active in Hymenoptera and honeybee research community. And so for over 15 years, we've been running the bovine genome database and the Hymenoptera genome database. And both of those projects needed more efficient access of data. And so that's when we started actually developing these data mining warehouses. So we have Bovine Mine and Hymenoptera Mine. And then after presenting these projects in different places, we came into contact with people who were interested in using it, but they worked on other species. And so that's how we had the idea of, well, let's expand our idea of Bovine Mine to other livestock organisms. And then also, now that FANG data is being generated, let's modify Innermine so that it can actually be able to incorporate that kind of data. And I have to say that we are very thankful for all the work that Peter's group is doing on the metadata, because we try to take advantage of that as much as possible. And we've done a lot of work on modifying the FANG data model so that we can incorporate as much of that, the data, metadata and the ontologies as possible. And so we've also started developing MazeMine in collaboration with MazeGDB. And so that came about because we were presenting FAGMine at the Ag Biodata booth at PAG. And then we met a group from MazeGDB and said, oh, well, maybe we can develop MazeMine and have a similar kinds of tools for maize. So we've been doing work on developing MazeMine for about 5 years. And then we've also just now started working on Aquamine. So Aquamine will support aquaculture species, and it will be similar to FANGMine. So that's all I wanted to say on FANGMine. Now I would like to move on to something else. It's actually, I want to talk about a pending proposal, which I know is probably bad luck, but I think that by presenting this, our ideas for this proposal, then we may be able to get feedback. And if we need to submit again in the future, we'll have feedback from the community. But this is about a proposal for cyber infrastructure for animal genomics in the US. And so this is a proposal that was submitted to the National Science Foundation. And I'm showing here the team involved in the proposal. So then all the rest of the slides that I show are actually from our ideas from this entire team. And so we have 5 institutions: University of Missouri, Iowa State University, University of Arizona, UC Davis, and Oklahoma State University. And so our team has actually been working toward these goals for since 2017. So in 2017, we were asked to write a white paper on U.S. animal bioinformatics needs to the NSF. And so Weijun Zhu led that white paper. And so we submitted or provided a white paper, just a short 1-page paper. And a year later, we were informed that there's an opportunity that we should apply for because this might help us meet our needs. So that was a mid-scale RI1 infrastructure. And so that's when our team formed. We submitted a pre-proposal in January 2019, but that was not successful. And on talking to the program officers, they liked our proposal, but it just didn't make it. There was other proposals that were more competitive. Pre-proposals. So they suggested submitting for the Science and Technology Center. And so we turned it around. We actually formed a larger team at that time. And then a few months later submitted a pre-proposal for a Science and Technology Center. And that's a very large scale project. Unfortunately, that was not successful. And so another mid-scale project opportunity came up just recently last fall. And so we resubmitted after learning some lessons from the first 2 experiences. And our pre-proposal was actually successful. And so we were invited for a full proposal, and we did submit that in April, and we'll find out this summer if we're that successful. And so this proposal is about designing an infrastructure. It's not about implementing one, it's about designing. So our aims are to determine the research and training needs of the research community by engaging the communities in the design project, to address software engineering and workflow systems and infrastructure support challenges, and also to design an analytic platform to facilitate data integration, exploratory data analysis, and mining the genome annotations. And so I'm going to just talk about sort of some key features of this proposal. First of all, I just want to emphasize this is a design project. It's a 3-year project. It's really so we can determine the community needs and to develop some develop and test design strategies. And then that will allow us to develop a solid plan for a future infrastructure and then be able to develop a strong implementation proposal. Because this, if NSF funds us for a design project, they are not promising to fund us for an implementation of projects. So we need to actually come up with a very strong plan for implementation in order to seek larger scale funding to actually do it. And I want to say that an advantage of this being a 3-year project is that we'll be able to, during this time, coordinate with some of the things that Peter and Elisabetta talked about in Europe, so the Eurofang and NF Core and things like that, so that we won't be reinventing the wheel and we'll be able to actually collaborate as much as possible. So another thing about this proposal is that since this is NSF supported, we're required to support existing, our infrastructure has to support existing NSF supported research communities. And so what that meant is that we had to expand our scope so that we would, our project would actually support all animal research, not just domesticated animals or farm animals. And so we reached out to other animal communities who are generating epigenomic data, for example, insect species that are used in behavior learning and adaptation, marine species used in environmental epigenetics, species that are used in fundamental genetics and evolution. And so we have interest from those communities, and hopefully we have showed NSF that we have interest. So we really want to engage the community in design. So we're going to have booths at conferences, virtual forums where we can have discussions with the community, online surveys, prototype training workshops, both in person and online. We'll have a scientific advisory board. We'll also have a user working group, so larger than a typical scientific advisory board, but a group of people who will be dedicated to testing out things and providing feedback over the 3 years. And then we'll also have an external evaluation team. So we're going to leverage existing consortia to reach out to research communities. So I just listed those here, and there might be more in the future. So like I said, that we're interested in scientist training. So we need to identify the community training requirements. And then part of this project, the design project, is not just to design the infrastructure itself, but how to design our training strategies and what resources do we need in order to effectively carry out the training during the implementation project. So we will be holding prototype workshops just so that we can get user feedback and evaluations from the external evaluation team on the best training strategies. So when we hold these workshops, we'll be using just some commonly used epigenomic analysis workflows. So these would be workflows that are already in place, but they're not widely used yet. They're workflows that have already been used by FANG. And so we would plan to implement the prototypes and develop interfaces for those on Cyverse, and then use those in our prototype training workshops, and also be able to collect information on how the users interact with the interfaces, and then what are the barriers for use. And so really, we want to develop a plan to ensure that any future workflows will meet the community's use needs. So we also are going to develop and test workflow engineering strategies. So that's by prototyping these workflows using different implementation technologies, and then also on several different NSF supported HTC, HPC systems. We're also going to design and prototype a process for workflow quality evaluation and workflow evolution. So how do we evaluate the quality of our workflows, and how do we allow for workflows to continually to evolve in the future as technologies and algorithms evolve? So we want to design a metadata system and database schema to capture outputs of the primary data processing workflows. And then we're also going to test some of our solutions by prototyping a few workflows that are currently posing challenges for use by the animal genome research community. So another part of it is to develop a solutions for data integration and access. So something, FangMine, I feel, is a very small scale data mining tool. So what we would like is a scalable multi-omic database framework that allows all these kinds of workflow outputs to be integrated and allow for data exploration and integration and modeling and hypothesis generation. So that will include both, what kind of database do we need that would scale for that, and then also what kind of data interfaces do we need. So we plan to leverage existing NSF supported infrastructure, so Cyverse, XSEDE, or whatever the future replacement of XSEDE is going to be, Open Science Grid, Pacific Research Platform, and maybe others. And then here's a long-term vision of our infrastructure. So if we got to implement it in a future, we would like to develop a scientific gateway. So for now, we just called it AGAMOD, which is Animal Genome Annotation with Multi-Omic Data. That's mostly because we needed to call it something for the proposal, but the name of it can change. And so basically, this would be a gateway where users wouldn't really have to know exactly where their work is being performed, but there'll be a workflow execution service that will send out jobs to different NSF supported resources. And then there would also be a data mining and analytic resource. And then the idea is to be able to allow data to exchange between those. So that's all I want to say. And these are my acknowledgements actually for the FangMine project. And I apologize, but the acknowledgements for the proposal are on the previous slide where I listed all the investigators. And so I have, we have a couple of co-PIs, FangMine, Tom Spencer, Darren Hagen, and then we have several FangMine advisors representing different species groups who have provided input, and then a couple of former FangMine developers, and then current developers are Deb Tryant and Amy Walsh. And I just want to show Jack because he's our plant person. He works on MazeMine, and just to show that we're a cross-community lab. And that's all.
Okay, thank you, Christine. There's a question in the chat from Peter. Question is regarding AquaMine. What is the target data resource, and will it support data from Aquafang and from the functional annotation of all salmonid genomes, or FASG, and as well as other sources? So what are the plans for that?
So when we wrote the proposal, we had to, this is NIFA supported, so we really had to focus on US aquaculture species. But in reality, once we start doing it, we really want to incorporate that kinds of data, and that data will actually be able, I think, be available before the US data will be available. So so we will likely incorporate those. Mm-hmm. So I think we need to, the first step is to incorporate what we said we would do, and then add in those. But if that data is available before any kind of FANG kind of data is available for US agriculture, we'll prioritize, or we'll bump those up in the priority list.
Mm-hmm. Other questions, just put them in the chat or just shout out. I'll throw in a question. So it's interesting that all three speakers talked about, on the one side, the need to bring communities together, to train communities, but then also to obtain funding, right? And so obviously securing funding and capital involves being able to show the funders that you're able to capitalize and effectively use that funding and deliver on them. So how much of a part is that of all these initiatives, is to show funders that we have a community, that we are able to develop this, bring these communities together, and deliver on the funding that they provide? Any of the three speakers?
I can say that it's very important. And for example, I mean, just getting advice from the NSF program officer on submitting our proposal, that was a big thing. Like we had to show, we have letters of support and we had to show that what communities would be interested in. So I think it's a huge factor.
Mm-hmm. Peter or Elisabetta?
I agree with Christine. I think it's a big component. EU also put a lot of efforts in whatever is clustering of different efforts, like it did for these calls that I mentioned lengthily in my presentation, like the 3H 2020 projects, to really cluster together. Although, as the officer told us recently, usually this kind of recommendation turns out to be formally tick off in the deliverables you make, but not so seriously as the three projects are actually doing. And there is a massive investment in whatever is dissemination and training. Usually, it's really, these are really requirements. I think there is it's a real paradigm of research that is evolved and will not go back.
Mm-hmm. Okay. Other questions?
I had a question for Christine. I tried to write it, but I messed up.
Yeah, just speak up.
Christine, yeah, I mean, so it's very interesting about this cyber infrastructure that you have submitted, and of course, maybe we get it and it will be great. For the future, when there will be, we will start soon with Amélie Clark, the design of the infrastructure project for the EU. And there will be more of these projects. And I tried to say in my presentation that there should be a harmonization of infrastructures from diverse countries. So how do you see this? Where would you start for cyber infrastructures specifically?
Oh, you mean when we, like, how do we start interacting or?
No, I mean, imagine that you get your project and you start to make all the design that is, in essentially this proposal, in this project that you will start. And in the meantime, we will have to go through similar stages for building the EU proposal, and maybe in Australia the same and so on. So what would be the first step, the most important step you'd think to start with, to harmonize this kind of efforts together?
Like we really emphasize reaching out to communities at the very beginning of the project. So the whole first year it was, we have some meetings that we want to go to in booths, and then reaching out to communities in various ways. And then we will, there's only like a couple pipelines that we're going to put onto Cyverse just for testing. They're actually the UC Davis pipeline, the pipelines used in the most recent paper, the 2021 paper that you mentioned. And so I mean, I think that's the most important thing, is to actually just get input from as many people as possible. It will be the end of our 3 years before we actually we really know specifically what we need to do. So we have all these ideas. Of course, we have to propose something, but we need to continue to get input throughout the whole time and kind of evolve our plan. And then the NSF doesn't promise that they'll pay for implementation. So ideally, we'd be able to start implementing like in 4 years, but we don't know. So.
No, because just as an impression, my feeling is that when you talk more generally about communities and the needs of the communities, what people would say, you look at the topic calls for research, they actually want the same things. They're always the same. I can tell you that also in the EU, the new program for infrastructure includes as well a program for designing infrastructure, these not so big scale funding schemes. And we'll probably go for it. And the goals will be these big integrations with the keywords that are always the same. So sometimes I think that we may find ways to disperse a bit less, to be more focused, without spending time to use the same words in different combinations, but finally wanting the same things.
I think, a problem that we have, like for here, is that we've always had such a short turnaround time. When we find out there's a proposal, we only have 1 month to do it. Like, and then, you're scrambling to like reach out to anybody. And so it's really hard to, in the proposal writing stage, to be able to actually reach out and actually have a good plan ahead of time. I don't know how it is there, but NSF, so the mid-scale announcement didn't come out until late November. And of course it was like right around Thanksgiving when everybody's on vacation. And then it was due a little bit over a month later. And then of course you have holidays in between. So, but I think it's good we're talking now, because now like we know about it. We know about each other. So we have to do it in the future. We can, it'll be faster to communicate it.
I think it's very interesting because, first step is to get the funding to do things, right? But I think even if, and when you do that, you have to show that what you're doing is unique, right? And different from what others are doing. But I think it's good. Well, what we see happening is that once people get funding, there are discussions like here to see, well, how can we collaborate and how can we integrate it so that we all benefit from it? And that's fine, I think. That's the way the system works, right? Chris?
Thanks, Jack. Yeah, that's a perfect entree to what I was going to say is AG2PI has some funding for a conference or even perhaps multiple conferences. And I think one of the goals we had was to bring the plant and animal or crop and livestock communities together. So to try to solve some of these exact issues, or at least to start to work on them. And so I think that's a perfect indication to us that this should be one of the major points to develop that conference idea, is to bring the data resource communities together to make sure that they're interacting.
Okay, well, we're over our time, a lot of time already. So I think the Slack channel will be open. So if you have questions for any of the speakers, or you want to continue the discussion, please make use of the Slack channel. I want to thank Nicole Scott and Carol Brown, who helped with the technical details and getting the information out. And of course, thank our three speakers, Elisabetta, Peter, and Chris, for excellent presentations, giving us some good discussion material and insight on what's happened already on the animal side, and similar things are of course happening on the crop side. And integrating that will be a key for the future. Next month we'll have another field day every third Wednesday of the month. Training workshop will start June 24th. A practical guide to genome-wide association studies. So this is part of the effort of AG2PI to train the community, which is again common to what other efforts we've heard about today. And so again, there some integration would be very useful. So with that, I'm going to close it. So thanks again for everybody for attending, and have a good rest of the day.